Bonjour à toutes et à tous, bienvenue. Est-ce que les seuls Anglais que je vois dans la salle parlent français couramment? Et tu avais prévu de faire ton talk en anglais? Ouais. Mais je ne sais pas si moi je dois parler en anglais, j'ai l'impression. Is there any English speaker who is not understanding any French? Magnifique. Oh, on a même la directrice de l'ISPED aujourd'hui, c'est magnifique. Bonjour. Donc, d'abord, je vous souhaite à toutes et à tous une excellente année pour ceux que je n'ai pas encore vus ou écrits. Et j'ai encore quelques mails de retard par rapport aux vœux. Donc, bienvenue pour c'est le premier séminaire de l'année. Ouais, c'est le premier séminaire de l'année 2025. Et c'était le minimum pour Laurent. Donc, Laurent Billot, en fait, c'est un statisticien qui travaille au George Institute en Australie. Il nous en dira peut-être plus sur cette organisation. On a souvent rêvé avec notamment Christophe Souriau de réussir à faire quelque chose comme ça, qui est un institut, une organisation, une non-profit organization, mais qui est partie vraiment de l'académique, donc très ancrée en Australie. Alors maintenant c'est University of New South Wales, mais avant c'était Sydney au départ. Et en fait, la particularité, c'est qu'ils ont... donc, Laurent, lui, est responsable de la division Stat. Et en fait, depuis quelques années maintenant, il est physiquement basé à Bordeaux. Et donc, ça fait longtemps en fait qu'on collaborait ensemble. Il y avait aussi des collaborations historiques de Christophe Souriau avec le George Institute. Mais comme on était très contents de travailler ensemble et qu'il était prêt aussi à participer aux enseignements, on a pu le nommer Adjunct professeur dans le graduate programme Digital Public Health, ce qui explique que nous l'avons accueilli dans les locaux version ISPED pour être avec nous régulièrement. Et puis, comme d'habitude, on s'est dit c'est quand même pas normal puisque... en fait, voilà, quelqu'un qui travaille beaucoup, qui a une expertise importante et qui développe des choses autour des essais adaptatifs et certains travaux en collaboration avec nous. Et comme d'habitude, on ne sait jamais les gens qui sont ici ce qu'ils font. Et donc, on s'est dit, on va l'inviter, puisque en plus, avec ses casquettes, ça nous permet d'avoir un invité expert international qui va nous parler des essais adaptatifs. Donc, merci encore, Laurent, d'avoir accepté cette invitation et surtout d'être avec nous parce que ça nous renforce. Merci. Donc on va t'écouter, nous sommes tout oui.
Merci Rodolphe. Et puis, moi merci pour l'accueil, pas seulement aujourd'hui, mais depuis que je suis arrivé à Bordeaux. Je suis arrivé à Bordeaux en 2019 et en fait, j'ai toujours été accueilli tellement bien, je me sens ici chez moi, notamment grâce à toi Rodolphe. Et comme tu disais, donc c'est Simone qui m'a grillé dans les couloirs il y a quelques semaines, qui me dit... mais ça fait longtemps que tu es là, toi. Comment ça se fait que tu n'as pas fait de présentation au séminaire de l'ISPED? Bon, voilà. Je me suis dit, bon, on va faire quelque chose. C'est pour ça que je suis là aujourd'hui. Pas que, hein. Je suis content. Mais alors, avant que je commence, j'avais prévu de faire en anglais. Moi, je fais en anglais? Ok. Bon. Donc, je vais faire ça, alors. Donc, oui, bonne année. Bon, je connais most people. Ça y est, je change en anglais. But not everyone. So, thanks for the introduction. So, I'm Laurent Billot. I've been working at the George Institute for 20 years. That's based in Sydney. I was in Sydney for 15 years. In 2019, moved to Bordeaux. That's when I joined the team here. I spend like 1 day a week in the office upstairs. But otherwise, I work from home, travel to Australia, India, the UK, where we have offices, and my team is spread across all those regions. But mostly here, working from home or in the office for the, adjunct professors or invited academics. So, like Rodolphe said, most of my work, so I look after the stats division of the George Institute. So it's not for profit. It's affiliated with the University of New South Wales in Australia, Imperial College in London. So we have a few bases. A lot of what we do is around cardiometabolic, non-communicable disease. So everything that's not contagious, basically. Although we did work a bit about COVID, and we'll talk about COVID a little bit later. So cardiometabolic injury, critical care, it's quite broad, and most of what we do is based on large-scale clinical trials. So a lot of the work that I do and my team does is focused on clinical trials, hence today's talk. And what I want to talk to you about is not just clinical trials in general, but a bit more what's happening in the adaptive trial space and what we've been doing. So I'm probably preaching to the converted here, but why are we so interested in clinical trials? I guess probably most of you have seen this already. That's what we call the pyramid of evidence, where at the top you have randomized clinical trials as the strongest source of evidence. Why is that the case? Because in the clinical trials we randomize people who are exposed to an intervention versus those who are not exposed, and that presence of a randomized control group allows us to make causal inference. But clinical trials, they've been around for a long time, but they have lots of challenges. Some of those challenges are not new. Others are more recent, or at least we're more aware of them recently. Recruitment is always a challenge. Retention of participants, those are probably the two biggest challenges. And in fact, there's lots of work by groups trying to improve recruitment, retention. There's work in terms of study within trials to try and improve conduct of trials, SWATs, we call them. The time and cost, it takes a long time for some trials. I know that for some of the trials we're involved with, that the cardiovascular trials, for example, the last 5, 6, 7 years. I think there's the longest one, this trial I'm working on at the moment started in 2016, and it's due to finish in 2028. So it can take a really long time, can be very costly. And more recently, we've started to worry a bit more about other aspects, which is the engagement. I'll talk a bit more about what I mean by that, but I guess how we engage with the community at large, the trial participants, both at the design stage, but at the dissemination stage, and of course, the carbon footprint. And when we look at the cost of clinical trials and the carbon footprint of clinical trial, what we see is that most of the cost of healthcare or drug development is not due to clinical trials, but there's still a significant portion, it's 5% of the total. And that's what we see in the middle. I'm going to try and make this work. No, wrong button. This one? Yes. Here in the middle. So the cost of clinical trial is 5% of the total cost, and that's mostly driven by travel. Travel of participants, travel of staff to go to the sites. So you can see that there's a cost imperative, there's a duration, time, but it's also the carbon footprint. So the more efficient we are, the more we are going to reduce all those things, the cost and the carbon footprint of trial. There's more and more attention given to this, and we'll come back to this towards the end a little bit. But recently, the NIHR, so that's the main funding body in the UK, has issued some key recommendations for researchers in terms of the carbon footprint. And you see that when you look at the list, it's not just about directly carbon, but it's all the things that contribute to the efficiency of trials and are likely to reduce the overall footprint of the trial. And that includes having the correct research question, so we don't waste time and resources, having an efficient study design, study setup, conduct, unnecessary data collection. There's a lot of work around this. We tend to collect everything we can just because we have a trial and we want to maximize the information. That's actually potentially quite wasteful in terms of the time, burden, resources, storage, etc. Clinical trial monitoring, we saw on the previous slide that a lot of the cost is due to travel. So we're talking a bit more and more about risk-based monitoring, remote monitoring, but also good practice in reporting. You know, it's all good to do really good trials, but if we don't report it properly, so it's not implemented in practice, that's also a waste. And everything that's a waste contributes to that carbon footprint. And overall, reducing the environmental impact. In this presentation, I'm going to focus mostly on the efficient study design and the statistical aspect, and at the end, I will come back a little bit to talk about the other aspects. So that's still from the same websites. When they talk about the efficient study design, they talk about making use of resources, patients, populations, patient time, consider whether it's possible to answer several questions through one study. They talk about factorial design, but I guess that's the idea also of platform design. And we'll talk a little bit about platform designs. And making sure basically the design is done properly by involving statisticians, methodologists at the design stage. But let's talk about adaptive trials, because I guess what is an adaptive trial? It's really the underpinning idea is to make sure that the trial is efficient and that we get the answer to the question as quickly as possible by adapting as we go. There's several definitions, which is probably one of the most common from a working group on adaptive trials. And the idea here is that we use accumulating data to modify aspects of the studies as it continues, and importantly, without undermining the validity and integrity. So it's not like we can change anything by accumulating information. There's some rules. There's another similar, but I really quite like this definition from James Wason, where he talks specifically about improving the power and efficiency and the participant benefit of the clinical trials. And again, it talks about the fact that this has to be done in a statistically robust manner. So there's some rules. So I mean, typically, and I assume most of you are at least familiar with the overall trial. The overall or traditional trial, so, let's say, is the trial where we have some assumptions, we have a research question, we design it, we conduct the whole trial, and at the end we analyze and we find out whether or not we have the answer or we were able to show some effect of an intervention, typically. The idea of the adaptive trial is, as shown in this little graph here, is that we add those two bits at the top, which is that we review the data while it's accumulating and we give ourselves the chance to change things, to adapt, instead of waiting until the end and decide, oh, too bad, didn't have enough power, for example, or we could have stopped much earlier. There's very strong evidence. So that's the typical kind of adaptive trial, is deciding early that we have enough evidence to stop because it's already proven that it's working and we don't need to wait until the end, or vice versa. It could be the case that we look at the data, it's so, the chance of seeing some positive result is so low that we stop the trial for what we call futility. But there's more aspects to this. There's ethical aspects as well. So there's some types of adaptation where we might modify the treatment allocation so patients are more likely to receive the more beneficial, the better treatment. That's what we call response adaptive randomization. We'll talk a little bit about that. But the underpinning idea is really that flexibility to make modification as we go. But those modifications has to be pre-planned. It's just a list of some of the common adaptations that people do. A very common adaptation that people do all the time is sample size re-estimation. So reviewing the assumption that we've made at the start to check whether they hold still and change the sample size, mostly increasing the sample size, although many investigators would like to be able to reduce it, but that's very rare. Changing the trial duration, changing the follow-up, so it could be changing the recruitment, or it could be changing how long we follow patients that have already been recruited. Could be changing the endpoint, adding or dropping treatment arms, looking at specific subgroups, changing the inclusion-exclusion criteria to then enroll patients who respond more in a certain subgroup, or response adaptive randomization. One thing that's important to note is there's two families, I would say, of adaptation. There's the blinded kind, that's the dog on the left, and the unblinded. What I mean by that is a blinded adaptation is one where you make change, you review your data, but you don't review it by randomized arms. So if you have two arms in a traditional parallel two-arm randomized trial, you look at the data overall, like a cohort, look at the overall variability, you look at the overall rate of missing data, etc., etc. Based on this, you can do pretty much any adaptation you like. Because it's blinded, you're not going to introduce any bias, you're not going to inflate the type 1 error rate. People have been doing that kind of adaptation, reviewing the standard deviation, for example, etc., for a long, long time, and that's fine. And I'm not going to talk about those. What I'm really interested in is the unblinded adaptation, where we look at things by treatment arm and make decisions. And that's where it's important to make them in a statistically robust manner, because if you make decisions based on unblinded data without certain rules, you will inflate the type 1 error rate. You will introduce some bias. So here we're talking only about unblinded adaptation, and that's really what we mean when we talk about adaptive trials. So just a week or so ago, I just ran a search out of interest and for context on clinicaltrials.gov to look at the number of trials that mention adaptive designs or adaptive trials. It's probably not the best search, but at least it gives us an idea of what's been happening in the recent years. And you can see on the left, 2005, there was only one trial that mentioned adaptive design or adaptive trial recorded on clinicaltrials.gov. And it's been increasing steadily. But what's interesting also is that in 2020, 2021, 2022, there's been a huge increase in the number of adaptive trials and adaptive design. I'm sure you know why that's the case. And that's because of COVID. And what happened in COVID, there was a great sense of urgency that we needed to come up with interventions and enough evidence to be able to implement or not implement them as quickly as possible. And the answer was adaptive design. Why? Because if you adapt in the right way, and there was a lot of bad trials during that time, but the idea was to stop the trial as soon as there is enough evidence that an intervention works and can be used in practice, or that it doesn't work, which then allows you to deploy your resources to test something else. During that time, a lot of trials were done, a lot of new adaptive trials were done. Interestingly, probably the most well-known adaptive trials for COVID and overall is the one that's called REMAP-CAP. It was in place before COVID started, and the idea was to look at community-acquired pneumonia. It had lots of different domains. What I mean by domain, I'll just open a quick parenthesis, quick aside to explain what we mean by a platform trial, because the REMAP-CAP is a typical example of a platform trial. The platform, there's two general ideas behind the platform trial. The first one is that there's a whole infrastructure of sites, investigators, patients, so you don't have to finish a trial, reopen sites, reinitiate them, etc., etc. So you start building a network, and using that network, you can test not just one. You can run not just one trial, but multiple trials, multiple interventions at the same time. The idea of a domain is really different interventions. So in the critical care setting, which is where REMAP-CAP is happening, one domain could be the type of antibiotic you give to the patient who have pneumonia. Another domain could be giving them steroids, or it could be fluid resuscitation. So it's all the different types of intervention that you give to those patients, but you test them in a randomized fashion on a common platform. So instead of having subsequent trials, one trial testing steroid, you recruit new patients to test antibiotics, you finish that trial, you restart another one. You have one common platform, and the patients are eligible for more than one intervention or more than one domain can be randomized to more than one and be tested at the same time. It's a bit like a factorial trial, really, except it doesn't have to be completely factorial. Some patients will be eligible for antibiotics, others for steroids, some for both, etc. So REMAP-CAP started, I'm not sure exactly when it started, but it would have started around 2016 to 2017, I would say. So it was in place when COVID happened. It was in perfect position to expand and test intervention for COVID patients. And in fact, there's the numbers here at the bottom. It ended up recruiting almost 20,000 patients with COVID. REMAP-CAP is, one of the problems with REMAP-CAP, it's quite complicated. It's currently testing 66 different domains. So it's huge. It started with one or two, antibiotics, steroids. Now it's testing 66 different domains. The DSMB, so Data Safety Monitoring Board, meets every month to review things, to look at the Bayesian reports to see whether need to adapt the randomization, drop an arm, add an arm, drop it for futility equivalence. It's hugely complex. There's a great infrastructure, but it's very complicated. But I guess if we were able to do this, and if we felt compelled to do adaptive design during COVID, why don't we think it's a good idea to do this all the time? I just ran a search on PubMed, slightly different to the one on clinical trials. This is just for published protocol. What we see is a very steady increase in adaptive designs, with almost none in the early 2008 to, I'm not sure what the number is here, but big increase in 2024. I was pleasantly surprised when I ran this search to note that the latest articles actually are protocol. So the latest adaptive trial protocol published in 2024 is the Captivate trial, which I will talk about now. So the Captivate trial is both a platform and an adaptive trial. So let me pause here for a minute, because technically a trial could be a platform trial that is not adaptive, okay? Because the platform, like I said a few minutes ago, is really about setting up an infrastructure. So you could very well have a platform trial where you test lots of intervention, you have a common infrastructure instead of restarting one for every new trial, every new question, but you don't have to adapt. You could still have your traditional trial, factorial trial, based on that common infrastructure. In reality, most, if not all, platform trials have some adaptive component where there is some decisions to add an arm, drop an arm, change the randomization ratio. The reverse is also true. In fact, most adaptive trials are not platform trials. So the Captivate platform trial is both, even though we started with only one domain, and most adaptive platform trials only start with one domain with the idea to get more funding and have an infrastructure in place to attract more interventions, more partners. What we're looking at in this trial is patients with chronic kidney disease. And when you have chronic kidney disease, your renal function, which is measured by eGFR, declines over time. So our outcome, which is now accepted by the regulators, is eGFR slope, so the decline in renal function over time from randomization to week 108, so 2 years. Because it's 2 years and it takes a long time to observe, we're using a surrogate outcome, which is the urine albumin-creatinine ratio at 24 weeks. This leads to 3 types of rules. So remember, I told you earlier, typically we'll stop a trial for early success, so there's early signs of efficacy, or early futility. This is based on a Bayesian adaptive trial, like REMAP-CAP, so we're combining prior information with observed data, calculate the posterior distribution, and based on that posterior distribution, we make decisions about certain probabilities. So futility is based on 2 possible rules. One is based on the actual eGFR slope, our real outcome. The other is based on the urine albumin-creatinine ratio at 24 weeks. And this is saying that if the chance of the difference in the albumin-creatinine ratio, that difference being more than 25% in the intervention arm, so some substantial, interesting clinical effect, if that probability is less than 5%, we say, stop the trial. So we're saying the chance of having a meaningful effect is too low, so we stop the trial. Likewise, if we see that the difference, absolute difference in the slope, the chance of this difference being greater than 0.8, which is, again, a clinically important difference, if that's less than 5%, we stop for futility. Vice versa, if the chance of having a positive difference, so difference more than 0, is greater than 98.5%, we stop for efficacy. I can't remember exactly the schedule, but I think roughly we were planning to do an interim analysis every 6 months, calculate the posterior probability, update them, and stop for futility or efficacy if we cross those boundaries. At the moment, we're not using adaptive randomization. I will talk about this, but depending on the next domains, if we have multiple arms, we might. This is what it looks like. Sorry, it's probably hard to see. There's 1, 2, 3, 4 domains. At the moment, there's only one domain active because we only got funding for one domain. We're trying to get more money, applying for funding to get the next domain, but we're still waiting to hear the outcome. But the idea really is to have that common infrastructure, which is in place, get some funding from agencies, funding agencies, for industry partner, and test new domains on the same platform. The 2 examples I showed you were Bayesian trials, and they're actually quite complicated. There is, my impression, I don't think it's just me, that impression that an adaptive trial has to be Bayesian and therefore quite complicated. And in fact, a few years ago, one of my colleagues, we were planning some reanalysis of trials, and we were considering adaptive designs, including some Bayesian trials, some Bayesian design, as well as group sequential design, so frequentist design, which I'll talk about in a minute. And when I suggested a group sequential design, my colleague said, no, we really want to do an adaptive design. So, well, that's an adaptive design too. But there's that understanding, there's that impression that to be adaptive, it has to be Bayesian. And yes, it's possible to do it with Bayesian, but it tends to be quite complicated, and I'll explain why in a minute. And in fact, it seems to me that in most cases, we're able to achieve very similar things with a more simple, more established design using group sequential design or frequentist approach. This is an example of a protocol that was published, I think, last week for a trial of a pain medicine for people with sciatica. And, you know, I thought, why don't we go with a very simple adaptive design, a group sequential design? So what we did is we applied for funding, and we said we would do 2 interim analyses, so not too many, just 2, using very well-established group sequential design. So this picture on the left is showing the boundaries. This is the Z value, the test statistics on the y-axis, showing the boundaries to stop for efficacy or harm. They're symmetrical. So if one of the 2 arms is better than the other, we would stop for efficacy or stop for futility. Basically, this is saying that if the test statistics at this stage of the trial is very close to 0, the chances of it moving to that zone at the end is so low that will stop for futility. So that's a very well-established design. It's called O'Brien-Fleming. It's been around for decades. What we showed doing this, we did some simulation, is that based on 10,000 trial simulation, we are able to save 58 participants, which is 22% of the sample size, compared to a fixed design where we don't do any interim analysis. The other thing that's interesting here, initially, in fact, I designed it with 3 or 4 interim analyses. We applied for funding, we got the funding, then we wrote the protocol. My collaborator said, well, 3 or 4 interim analyses, that's a bit difficult logistically to run them. Could we do 2 and keep the sample size? I said, no, I don't think we can, except that with some work I'm doing with a research fellow at Imperial College, we found that if you change the timing of the interim analysis from the typical even spacing, so typically if you do 2 interim analyses, they'll be at 33% of the patient and 66%. If you move them to 50% and 70%, you save enough sample size that you can drop interim analysis. So that's what we did. Currently, we're working on a paper which is looking at optimizing the timing. But basically, what it's saying is do them later. There's almost no reason to do them before 50% because the chances of stopping, unless you use very liberal stopping boundaries, the likelihood of stopping is so low that you're just wasting resources. You might be better off doing them later and saving on the sample size. We also did a reanalysis and simulation study where we compared lots of different adaptive design, both frequentist and adaptive. That's when my colleague told me that frequentist is not adaptive. And this was a reanalysis of a previous trial. So the adrenal trial is a trial in critical care where we gave steroids versus placebo, randomized around 4,000 patients, 3,800. And it was designed to detect a difference from 33% in mortality at 90 days down to 28% with steroids. And we planned 2 interim analyses with Hebital-Pito boundaries, no futility. Hebital-Pito is constant boundaries for efficacy, there's no futility. And the sample size was 3,560. The question we asked ourselves during that, when we did that work and wrote that paper is, what if we reanalyzed it, we had designed that trial with different stopping rules, with futility, more frequent interim analysis, and Bayesian designs. I won't share all the details, but what we found is even with very simple Hebital-Pito boundaries, just the fact of increasing the number of interim analyses has a dramatic impact on the expected sample size. So this is the sample size if we don't do any interim analysis, 3,560, which is this. I should mention, because in case you're confused, even though at the design stage we said we would do 2 interim analysis, this was not adjusted for. So we just did a sample size calculation, assuming no interim analysis, came up with a sample size as a fixed design, 3,560, and people happily at the time, not so much now, completely ignore the fact that we were doing interim analysis, the impact on the expected sample size, the impact on the type 1 error rate. Until very recently, and I still have some colleagues who say, hey, Peter, it's very conservative, we don't need to adjust anything. Well, that's not quite true. But that's why our sample size was 3,560, which is the sample size with no interim analysis. What we see is, if we do up to 5 interim analysis, which for a trial of that size, which lasted several years, is not that extreme. It's like 1 interim analysis a year. You save 23% of the sample size. It's huge. But the maximum sample size, which is the black curve, you have a penalty because of multiplicity, but only increased by 2%. So the trade-off, you can see, is very, very beneficial. And that finding is very common, regardless of the type of boundaries, whether it's adaptive frequentist or Bayesian. That's the kind of finding you get. Let me just have a. No, it's gin. So that's another trial in the critical care space that we designed recently. It's called Prevent Neuro. And again, the outcome is 90-day mortality. We're expecting in the control arm to have 38% of mortality. The trial was powered to detect a 5% absolute reduction in mortality, so that's a relative risk of 0.85, 90% power, fixed frequentist sample size, 2,946. I wasn't aware of that trial until my colleagues came to me and say, oh, we'd like to redesign this as Bayesian adaptive trials. I was like, okay, why Bayesian? Oh, I don't know. Sounds cool. I couldn't really convince them. Half of them were like, we don't care if it's Bayesian or not, and 2 of the investigators really wanted it to be Bayesian. So I said, okay, well, let's do Bayesian. So we run some simulations and redesign it with a Bayesian adaptive design using those parameters. So, oops, sorry. So one change, because we added futility, we switched from a 2-sided alpha of 5% to 1-sided alpha of 5% with a futility boundary, a non-informative prior, and we decided that we will review efficacy after every 300 participants. And if the probability that the relative risk is smaller than 1, so if there's an effect of the intervention, if that probability is greater than 99%, so there's a 99% posterior probability of success, we stop for efficacy. For futility, the team was worried about stopping too early. So initially, we designed it with the same frequency. Every 300 patients from 0, 300, 600, we would test for futility. We came up with a sample size, but they said, oh, we don't really want to stop for futility early, so let's not do futility until we have at least half of the patients, of 1,500. And the rule here is, if the chance of having a meaningful effect, and that's the relative risk of 0.85, which is what we powered for, is less than 1%, we stop for futility. This took quite a few weeks of simulations, and we ended up with a sample size, expected sample size, so on average across all the simulation, that's what we mean by the expected sample size, of 1,588, which is great. I mean, that's almost halving the sample size. So the investigators were very, very happy with this. We submitted the protocol. I think it's under review or just got approved. I'm not entirely sure. You do get a little bit of a penalty, though, because of all the looks and the penalty on the multiplicity. The maximum sample size, so if you don't stop at the interim, you'll need to go to 3,300 to get 90% power instead of 3,000. But that's pretty minor increase when you think about the fact that we're halving the sample size. So we did all this. Everyone was happy. Took us many weeks of simulations. And then, actually, as I was preparing this presentation, I thought, but what if we'd done it with a frequentist design? It turns out, so I tried two common frequencies design. The O'Brien-Fleming, that's the one I showed you earlier, and the Pocock. And Pocock himself will tell you, don't use the Pocock design and stop calling it Pocock. That was a mistake. Yet, we all talk about Pocock, and we always look at the properties of the Pocock design, so I did. The Pocock design, as you can see, is, by the way, the y-axis is not the same on the both graphs, so sorry for this. This is much wider, which means that those stopping boundaries on the O'Brien-Fleming are much more conservative. It's really hard to stop at the beginning. The Pocock, however, see, this is a value of 2, 3 here, so it's not that far from the nominal value for a test, a 95%, 5% significant test. So it's much easier to cross the boundaries at the beginning, which is one of the main criticisms of the Pocock boundaries, by the way. But anyway, I tried those two with the efficacy and futility boundary asymmetrical, a bit like the Bayesian design, really. And what I found is, so there's two graphs here. The first one is the probability of stopping for efficacy under H1. So that's the power, basically. And here, that's the probability of stopping for futility under H0. So that's what you want to get. If you're under the alternative, you want to stop for efficacy. If you're under the null, you want to stop for futility. What's very interesting here is you can almost not distinguish the boundaries or the probability of stopping between the Bayesian adaptive design, which is the orange curve, and the Pocock, which is the blue curve. They're almost exactly the same probabilities of stopping. The futility is a bit different, partly because with the Bayesian design, we didn't start testing for futility before we'd recruited 1,500 patients. So I didn't put that same constraint for the group sequential design. But I guess what's very striking to me is how similar those are, and actually the fact that these are not very different anyway. And with a bit of shifting of the probabilities, they could be very, very similar. The way they follow the same kind of curve. When we look at the expected sample size, so if you recall, under H1, the original design, so no adaptation, was just under 3,000. Bayesian adaptive design, expected sample size of 1,588, with a Pocock 1,521, almost exactly the same. In fact, a bit less than with the Bayesian adaptive design, except it took me 5 minutes to design it instead of 2 weeks. O'Brien-Fleming, you might recall the curves, they're much more conservative, much harder to stop at the start, so we have a bigger expected sample size. For under the null, it's a bit different. With the Bayesian adaptive design, because we don't start testing for futility before we have at least 1,500 patients, we don't stop as much. But overall, the findings are pretty consistent. The question then becomes, why should we use Bayesian designs? Can we do the same with frequencies design, which are much more simpler to design? There's very established formulas. Most software will allow you to do this. Had a look. There's not too many people who have been interested in that question. There's that one exception, which is that very good paper from Nigel Stallard, which compared Bayesian and frequencies designs, group sequential design, interestingly concluded that both approaches can have identical stopping rules depending on the critical value you stop for the probability of stopping. And then it goes on and says, basically, when you take an O'Brien-Fleming design, it's the same as a Bayesian adaptive design with very conservative, like negative prior information. And Pocock is the same as a Bayesian adaptive design with non-informative prior, which is exactly what we did for the Prevent Neuro, was a Bayesian adaptive design with non-informative prior, which is not surprising then. We found exactly the same probability of stopping than with the Pocock. One thing they say, though, is, and that's true, and that's probably the main advantage of Bayesian design, they are much more flexible. For example, it was very easy for us to say, let's not test for futility before we have at least 1,500 patients. We simulate everything, which is both a bad and a good thing. It's very time consuming, but because you have to simulate everything, you can test any scenario you like. But I suppose, and that's what they say here, there's nothing preventing us from doing simulation for the frequencies design as well. And probably we should be a bit more aware of what we are modeling. But I guess the bottom line is they're both equivalent depending how you tune the parameter. There's another article, and Laura, you might recall this one when we worked with Joyce, comparing a Bayesian frequentist design. When I saw this published last year, I thought, oh, that's fantastic. Except what they did here is they did a frequentist design where they tested for efficacy and futility after 500 patients, 700, 900, and so basically every 200 patients from the 500. But with the Bayesian adaptive design, they tested after every 100 patients, so twice as often. Surprisingly, not surprisingly, they found that the expected sample size with the Bayesian design was smaller. Well, yeah, you test more often. So that was not a particularly useful article. And that's pretty much it. There's very little in terms of literature comparing the two methods. The problem, as I said, is, and that's the main criticism, the Bayesian adaptive design rely on a lot of simulations. And so far in our institute, we've been relying on Berry Consulting. So Berry Consulting is based in the US. They've established themselves in the worldwide experts in Bayesian adaptive design, have a bit of a monopoly. In fact, they are the ones who helped us design the Captivate trial and another platform trial that we have ongoing and another stroke trial platform. And we're trying to wean ourselves from them and do the things ourselves, which we did for Prevent Neuro. Thankfully, as of October, for those of you who were at the International Clinical Trial Methodology Conference in Edinburgh, a group of people released the BATS trial, the BATS package, our package, which allows us to design Bayesian adaptive trials without extensive simulation using some approximation. Works really well. In addition, there's also a package, the equivalent for group sequential design that's called RPact, which both has an R package, also a Shiny app, which is perfect for people like me who don't know how to code in R. And that's the one I've used. No, I don't know how to code in R. I have very good people in my team who know. And that's the one I use to redesign the Prevent Neuro. It takes a few minutes. You can select any boundaries, any number of interim analyses. It's very, very well done. So at least it's becoming a bit easier to use them and to compare Bayesian versus group sequential trials. Okay, well, I have a few more slides just to conclude. The question I guess we can ask ourselves is, should we use adaptive design all the time? Are they always useful? Because it seems quite promising. And James Wason wrote a really good paper answering that question and looking at what are the situations where a Bayesian adaptive design, or no, not a Bayesian, but any adaptive design could be useful or not so useful. What it tells us is to be aware of three main things. The first one is the long-term outcomes. If you have a trial that takes a really long time to observe the outcome, by the time you adapt, that's too late. So an example, ideally what you want is a trial that recruits pretty slowly, but where you can observe the outcomes very quickly. The critical care space is not bad for that reason, because most outcomes are observed at 90 days or 3 months. So you don't have to wait very, very long before you have enough data to do the analysis and your interim analysis and adapt. The cardiovascular trials are more problematic because typically it takes years to observe a stroke, a myocardial infarction, or cardiovascular death. And if it takes 3 years to have enough data to do the adaptation, you may have reached a point where you've already recruited all your populations, and any adaptation is kind of pointless. So how long-term your outcome is, and that's why we use a surrogate for the Captivate trial. That's really important in, I guess, the scope, the ability to actually have an impact with your adaptation. The other thing, and that's a common criticism of adaptive trials, is the lack of information if you stop early. So a lot of detractors will say, yes, but if you stop early, we won't have enough information about secondary outcomes, or we won't have enough information about safety, or the evidence won't be convincing enough to change practice. So depending on the context, those are very valid concerns. I guess that's for us to assess whether we can still adapt and stop early, or we really need to have information on all those other things to really inform practice. And the last thing really is administrative logistical complexity. REMAP-CAP has an interim analysis every month, and that's a huge burden. One of my close collaborators is on the DSMB. He complains a lot about the REMAP-CAP monthly reviews. So what James Wason recommends is, first, there's not one method that's going to work for every case. So he thinks adapting is a good idea and that we should always consider some kind of adaptation, but there's not going to be always one design that works the best. Maybe it's going to be Bayesian, maybe it'll be frequentist, maybe it'll be O'Brien-Fleming, maybe it'll be 2 interim analysis, maybe it'll be 10. It'll depend. And we need to be very aware of this and don't make assumptions. Test, talk to our collaborators to see which one is the best one. And think about the balance of the complexity versus the adaptation. If you do an adaptation that's going to save 1% of your sample size, but then it's going to cost another 20% of the budget because of the burden, that's clearly not worth it. Very briefly, I wanted to mention, so I've talked a lot about adaptive design in terms of stopping early for efficacy and futility. That's not the only way to have statistical adaptation. These two things are quite popular and gaining in popularity. One is response adaptive randomization. And the idea of response adaptive randomization is as you accumulate data and see that one treatment is doing better than others, you change the randomization probability. So for example, if you have 3 arms to start, you randomize 33%, 33%, 33%. If arm A is shown to be better based on the Bayesian posterior probability of success, for example, at the interim, you change the probability so you start allocating more patients. So you might change the probability of allocation 50%, 25, 25. It's very appealing on the surface. People really like it, it's intuitive. It seems more ethical to give more chance to patients to be randomized to the more promising arms. It's very complicated statistically. I went to a presentation in April at Imperial College where a group of people including Sophia Villar, talked about response adaptive randomization. I was very excited thinking I will go there and I'll know how to do it. I'm going to use it. I came out thinking, no way. Still very complicated. They always have a solution. There's lots of potential problems. They always have a solution, but it's really hard for a common mortal to know which one to apply when. So it's a work in progress. I think this promise has a lot of appeal, but it's quite complicated still. I'd be keen to hear if others have a different experience. The other thing that we've started to pay a bit more attention to is what it's called promising zone. That was introduced by Mehta and Pocock a few years ago, in fact, but hasn't been used much. The idea is you do an interim analysis. If your results are promising but not quite there, then you have the ability to increase your sample size to have enough power to detect that result. It's a bit counterintuitive. You think, how does that protect the type 1 error rate? But in fact, they show in this paper, if you follow a certain rule, it doesn't inflate the type 1 error rate. It allows you to increase the sample size. And in fact, we just designed a new trial. It's called late MT. So it's removing clots in the brain after a stroke, ischemic stroke. And what we've done is combine a group sequential design using O'Brien-Fleming efficacy futility boundary with an unblinded sample size re-estimation using the promising zone. And what we're planning to do, we have not yet done that interim analysis. At the last interim analysis, so at this one here, where we look at futility efficacy, if we don't opt for efficacy or harm, and if we're not in the futility zone, but I guess in that promising zone, we will increase the sample size so that the conditional power, the power at the end of the trial, is at least 80%. So that's the idea. I have yet to do it. I'll finish on 2 things. So I've focused this talk on adaptive trials, which is mostly statistical adaptation. But if you remember my slide at the start, it's not just that. There's a lot of waste in trials because of what people call uninformative trial. There was a really interesting article published in 2019 in JAMA who talks about uninformative trial. Informative trial as in the importance of the question being tested, as in the proper design. I guess that's where we can talk about adaptive design, the feasibility, the integrity, and the reporting. The reporting is quite interesting, and in fact, there's several aspects to the reporting. I'll talk about that in the next slide. The other thing that needs to change is probably the way we include patients. And there's a very good document or page on the NIH website, so in the US, about inclusion of women and minorities and including people of different ages. When we look at the page on the inclusion of women and minorities, it says that there's actually a Public Service Act which mandates the inclusions of minorities and to make sure that the research findings are generalizable. Also talks about the fact that now every application is reviewed and the reviewers decide whether it's acceptable or not acceptable in terms of inclusion and representation. So when you talk about adaptation, it's not just adaptive design, dropping, adding an arm. Trials need to change to include the right people. They also need to change in the way we disseminate our results. We tend to be very good at disseminating our results in the peer-reviewed literature, but do we communicate our results to trial participants? It turns out not really. So in this paper, Kathy Gillis and her team looked at intention of communicating results and the reality of communicating results with participants. Short stories, it's very badly done. Oftentimes, the people don't even mention an intention, and even if they have the intention, they don't really do it at the end, or they don't say how they've done it. Seems to me, at least, if I were a trial participant, I would be really upset if I'm not told the results at the end. And there's a page on it's a CT toolkit, which says, I really like this quote, that informing participants of results acknowledges their contribution, shows respects and sees them not simply as a means to the researcher's ends. I'll finish with this slide because I can't really talk about new future trials without talking about artificial intelligence. And again, I think it was a few months ago, 2-3 months ago, a really nice paper was published about what's the place of artificial intelligence in cardiovascular clinical trials. And the reality is not just cardiovascular trials, it's any trial. And they talk a lot about efficiency in terms of automation, automations of adjudication of outcomes, automatic coding, etc., etc. So it's clearly, when we talk about adapting, there's a lot of scope also for AI. So our trials are going to adapt, are going to perish. So I have this picture of a virus. The virus constantly adapts. And then you have the crocodile, which hasn't changed for thousands of years. So are we here or are we here? Are we able to stay with the crocodile and be fine, or do we need to change constantly? I'd say it's probably a bit of both. The crocodile is pretty good. Yeah. It's pretty robust. And I think a lot of the basis of our clinical trials are a bit like the crocodile. We don't want to change the randomization. There's lots of principles we need to keep. But given the burden, given the cost, given how we've been engaging or badly engaging with patients, we do need to change. So I quite like what James Wasson says when he says, we support adaptive trials, we need those changes, but there's not going to be one size fits all. So I think what we need to do is be much more aware, test different scenarios. I'm talking here about the statistical adaptation, but also be mindful that it's way broader. It's way broader in terms of how we engage with patients, how we disseminate, how we engage with the design, etc. I'm aware that I'm starting to run out of time, so I'll finish here and thank you for listening.